Did you know that light can have a huge impact on our body and health? Not only does it influence when and how well we sleep, but it can also affect our mood, alertness, and even our behavior. Welcome to Light O'Clock. In this podcast, we unpack the science behind body clocks and how light influences our daily biological rhythms. In each episode, we chat to experts in the field and have them break down the scientific topics so you can be enlightened. In the last episode, we embarked on a journey following light from the environment all the way to our retina and brain to learn how our eyes react to light and how we can make images out of this. This week, we talk about this third cell type in the retina, which had long been neglected, the iPRGCs. And for this occasion, we'll be asking the question, how do our eyes use light to tell time of day? And we're joined by Rob Lucas. Rob is a professor of neuroscience and director of the Center for Biological Timing at the University of Manchester. Here, his research group explores how mammals can use light to see things and to tell time of day. Rob, welcome to Light O'Clock.
It's my pleasure. It's a great pleasure to be here.
So in the first 2 episodes of the podcast, we described how we have these endogenous rhythms, 24-hour rhythms. They are synchronized to the environmental light-dark cycles, and how light is really important for this synchronization. So light traveling to the eye gets to our suprachiasmatic nucleus, which is this little structure in our brain. And this sort of synchronizes the inner rhythm with the outer 24-hour rhythms. Then with Midel, in the last episode, we were talking about how light also travels through the eye, gets to the retina, right? To these rods and cones that we have there, and enables us to see things. So one question that comes to mind immediately is, how can the eye do both things? So enables us to see things, so make images of the world around us, see colors, but also enable us to synchronize with the environment. Is this also part of the role of rods and cones, or is there something else?
So that's a really interesting question. I think maybe it's helpful to take a step back, if you don't mind, and think about that job of telling time of day from a wider biological perspective, because then it can help us get back into the retina as we need to for humans. So, of course, the need to tell time of day is not unique to humans. It's not unique to mammals. It's not unique to animals. Almost all life on Earth has to tell time of day in some way or another. And so that requirement to measure light as a signal for time of day exists long before eyes exist in evolutionary history, long before there was a retina. And so the task of measuring light to tell time of day really doesn't have to happen in the eye at all, right? And you can see that obviously plants do it, fungi do it, even single-cell organisms of all types, many types do. So you don't need an eye in order to tell time of day by measuring light. And indeed, even when you move into animals, it's generally the case that animals with well-developed eyes still have photoreceptors, light-sensitive cells outside of their eyes that they use to tell time. So you're right to ask the question, because what that tells us is that, let's say, a bird with incredibly good vision from its retina also can tell time of day, but using a totally different light-sensitive mechanism that's not in the eye at all. And it's a peculiarity of mammals, of which, of course, humans are a member, that we, in our evolutionary history, lost all of these photoreceptors outside the eye, and so we've concentrated it all into the retina. It follows that if you think about it, we have gone from a situation where our remote ancestor had rod and cone photoreceptors in the retina for allowing us to see, and a photoreceptor somewhere else that was telling time of day. And that photoreceptor telling time of day has moved into the retina. And so for a long time, what we knew was that in mammals, this light detecting task, unlike in birds, was restricted to the retina. And so it was kind of assumed that this would be, therefore, the rod and cone photoreceptor, because if you to a first approximation, if you ask what cells in the retina are light sensitive, it's the rods and cones, right? It has to be that. But it turns out that no, this photoreceptor responsible for telling time of day has moved into our retina and has found a new home in a very small subset of cells in the retina, which are dedicated to this task of, one of the things they do is to measure light to tell time of day. So actually, the retina has three types of photoreceptors. We have the rod photoreceptors, which are responsible for dim light vision. We have the cone photoreceptors, which are responsible for daytime vision and allow us to sense color. And then we've got a third type of photoreceptor called melanopsin. And that melanopsin photoreceptor, one of its tasks is to measure the amount of light that's in the environment as a way of telling us what time of day it is.
Okay, very interesting. So we have sort of another type of cell, right?
We do.
How is it called?
So the cells that contain melanopsin are called retinal ganglion cells. So I'm not sure what you had heard earlier on in the podcast about how the retina works, but very simply, light is coming in through the front of the eye and it's falling on the rod and cone photoreceptors. And as I say, they're the most common light-sensitive cells. So what they do is they absorb that light and they translate it into a biological signal, if you like. And that biological signal then gets transmitted to a group of cells called retinal ganglion cells. And the retinal ganglion cells are the cells that form processes that form the optic nerve. So obviously, when we see with our brain based on signals generated in the retina. So those signals generated in the retina need to be sent to the brain, and the retinal ganglion cells are what's responsible for that. So if you like, what would be an analogy? If you think about a fiber optic taking information around the world, really, the retinal ganglion cells form that fiber optic to take the information from your retina and present it to the brain. And so the vast majority of the retinal ganglion cells in the human retina are relying on signals from rods and cones, and they're generating that information, and then they're just transmitting them onto the brain. But there's a very small percentage of them that have their own way of detecting light. So if you like, they can generate information from the light falling on them thanks to melanopsin and also signal that to the brain.
Okay. And so how are these cells different compared to rods and cones? Do they look different? And do they respond to different light?
Yeah, so they look very different. So one of the things about the rod and cone photoreceptors, particularly rods actually, is that they need to be very, very sensitive to light. So if you think about how difficult it is to find an environment that's totally dark, we can see, especially if you're dark adapted, very, 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 very dim light is enough to enable us to see something. And that's because our rod photoreceptors, for example, have lots and lots of protein in them that's responsible for absorbing light. That's called rhodopsin. And the structure of the rod photoreceptors is designed to have a region called the outer segment, which is like a long, if you think about a top hat at the top of the cell, and that top hat is just chock full of this opsin that's absorbing light, which means that it's got a very, very high efficiency for absorbing light. Cone photoreceptors have less efficiency of absorbing light, but they still have like a mini version of this top hat with lots of their light-sensitive protein. The retinal ganglion cells that contain melanopsin don't have anything like that. And actually, that's quite important because the way that the retina is organized is that light comes through the retinal ganglion cells before it hits the rod and cone photoreceptors. So you don't want retinal ganglion cells that absorb lots of light because otherwise they would shade the rod and cone photoreceptors. So they're really not connecting very many photons, which means that they're nowhere near as sensitive to light as the rod and cone photoreceptors. But for the tasks they perform, that's not necessary, right? Because they're working under daylight conditions or at least twilight conditions when, actually there's a lot of light around. So they don't need to have that very sensitive, high threshold sensitivity.
That makes sense. We mentioned before that for our circadian rhythms and internal clock, certain wavelengths of light are particularly bad. Is that because the melanopsin are responding to these specific wavelengths?
Yeah, that's right. So every photoreceptor has a particular range of wavelengths of light that it's most sensitive to, right? And this is just the chemistry of light absorption, that we can't. You get to a kind of wavelength where their sensitivity falls away, and they're very insensitive. One of the ways you can experience that is that people often use red light to maintain dark adaptation. So in circumstances where you need to retain good light sensitivity, let's say you're performing some task at night and you're out in the wild, then if you also want to have something where you have more high acuity vision, you can use a red light. And the red light will give you relatively high acuity, so you could read a map or something like that. But it won't stop you also being able to see in the dark, in the dim light when you turn the torch off. And the reason for that is that red light is not very efficient at activating your rod photoreceptors, which is what you're using mostly to navigate at night. So it's a really good trick. People should do it if you ever find yourself in that circumstance. So what that tells you, though, is that red light is activating a different photoreceptor more than rods. And that different photoreceptors are our cone photoreceptors. So we have cone photoreceptors are more sensitive to red light. So red light is in that scenario is preferentially absorbed by cone photoreceptors. So melanopsin, of course, then has its own range of wavelengths that it particularly likes. And the wavelengths it particularly likes are in the kind of blue part of the spectrum. So the peak is around 480 nanometers, which we see as kind of cyan, something like that. So it's bluey green, really, for most people. So that's when it's most sensitive, but it will still absorb green light, orange light, yellow light, even out into red light if it's very bright. So it's more a matter of how bright that light needs to be in order to activate melanopsin, rather than it just simply not being able to activate melanopsin at all. But yeah, that blue part of the spectrum is most efficient at activating melanopsin.
One question that comes to mind when we think about these two different roles of light, telling time and making us see things, is what about blind individuals? Mm-hmm. So we know they cannot see things, they cannot make an image of the world around them, but can they still synchronize to the light-dark cycle outside?
Yeah. So of course, there's many different kinds of forms of blindness, almost as there are people that are blind, because it's often a progressive condition for people. So they'll go blind at different rates and reach different stages of blindness. And most people that are blind will have some rod and cone function, actually. It's relatively rare to have those people who are profoundly blind that lack everything. But what you can do experimentally, of course, is you can generate models of blindness in, for example, lab mice. And if you do that, you can get rid of all the rod and cone photoreceptors. So this is an animal that would be classified as completely blind. They still have melanopsin, and they still do perfectly well at synchronizing their circadian clock to a light-dark cycle. I actually have a wide array of light responses which rely on melanopsin. So it's definitely possible then for a mammal without rods and cones, but only having melanopsin, still being able to synchronize their circadian rhythms. Then you look at the range of people suffering from blindness around the world, and you find a similar range then in the degree to which people have circadian rhythm disruptions. So there are people that are very profoundly blind who nonetheless are able to synchronize their circadian rhythms to a light-dark cycle. And there's a very famous experiment done in Harvard by the Seisler group, looking at people who couldn't tell you whether the room lights were on or not. And some of them, although they said they were unable to detect light, still showed suppression of the hormone melatonin by light, so completely unaware that they're responding to light. So it is possible then to separate those two tasks in blind people, that you do find people that are lack visual perception, really, because of the lack of rods and cones, but still have melanopsin and still have that circadian synchronization.
Going back to something you mentioned before, you said that we sort of discovered these cells pretty recently compared to rods and cones, right? And you also said a little bit about how we are uncovering the wide range of roles that these melanopsin cells have. Do we maybe want to talk a little bit about all these roles of melanopsin? So it enables us to synchronize, let's say, to the outer world. Does it also have other roles?
So I'd almost put that role of synchronizing our circadian rhythm to the light-dark cycle as part of a general task that it performs, which is measuring light to tell time. So that's partly about synchronizing our circadian clock. It's partly about the effect that light has on making people more alert. So this is when it's daytime, you should be more alert, and therefore you get this kind of override effect. Regulating our hormone axis, so there's production of melatonin, for example. And then there's kind of growing evidence that it can, in a similar vein, modulate impacts of cognition, for example, your ability to concentrate, and just generally driving you more towards that kind of daytime awake and doing things kind of state in many ways. So I'd put it like that. So that's one kind of set of things that it does. The other thing, having talked about the separation between vision and melanopsin, it also helps us see, which is a peculiarity that I think can end up being, if we're not careful, confusing, because it's not our primary modality of vision. But, for example, there's now pretty good evidence that it's one of the mechanisms we use to judge the brightness of places. So if you're judging how bright your room is, maybe melanopsin's contributing to that aspect of perception.
They're basically super cells, right?
Yeah, I mean, it's ludicrous because, as you say, only discovered relatively recently. It's about 20 years ago now. Certainly 21 years ago, most vision scientists in the world would have said, well, this is total nonsense. We all know that everything starts with rods and cones. So it's relatively new that we found them. And that's because there's so few of them compared to rods and cones. But they have an outsized contribution to our sensory abilities, to our light sensation. I guess it's a lesson in so many aspects of life that just because there's not very many of something and it's rare, don't mean it's not important.
Yeah.
So keep looking. You might think you understand something, but there might be something you've totally overlooked, which actually could be, just because it's rare doesn't by no means mean it's not important.
Also talking a little bit about how we can apply the knowledge that we have now about what these melanopsin cells are doing to think about our built environments, mm-hmm. right? So for the longest time, we only knew about rods and cones. Mm-hmm. And so we never really thought about light as something that was really changing our inner rhythms. Mm-hmm. And so we're also moving towards thinking about like lighting in architecture, right? And yeah, maybe lighting in the offices is not optimal. Lighting in the hospitals perhaps not optimal. Where do you see this going?
I think it's a really, really important point because, when you look at lighting design, architecture, and so forth, overwhelmingly now people design light because of its aesthetic appearance, and also from an engineering specifications perspective, to hit a target to allow us to see, comfortably. But if you think that there's another whole way that we're using light, it means that those targets aren't necessarily appropriate. And definitely something like, melanopsin during the daytime probably needs much more light than we're used to thinking about as a minimum for a built environment. Conversely, at nighttime, actually having enough light in order for us to be able to see very clearly could actually be a bad thing, right? Because it's going to be activating melanopsin to a certain extent and therefore shifting your clock. So I think it's just a fundamental change to thinking about what's appropriate lighting. First of all, that the right amount of light is time of day dependent. So probably during the daytime, you want more light than you think you want. And definitely in the evening and at night, you want less light than you think you want. So thinking about that. Then the other thing that you can do is you can start with lots of new lighting technology now to mix different wavelengths of light in different interesting ways to control melanopsin. So those kind of new lighting devices are coming down the line. What I'd love to see is something on the packaging of lighting that gave you an idea of how efficient it was at activating melanopsin. So at the moment, what you now get on the light that you buy it from the hardware store or online or wherever, it gives you its output in lumens, which is how bright it appears for us, but it's not related to melanopsin, or at least indirectly related to how bright it is for melanopsin. And it might give you an energy efficiency. It'd be really nice to be able to know how efficient that light were for melanopsin, so you could decide for yourself whether you wanted it or not, and what time of day you want to turn it on, and things like that. The other really, really important thing is to do with how we quantify light and therefore how we regulate it. So because melanopsin is responsive to different wavelengths of light to our cone photoreceptors, it means that when you calculate the brightness of a light, you need to weight the wavelength energy differently than if you were going to do that to predict cone photoreceptors. And so one of the really important things that happened over the last decade has been acceptance of ways of measuring light that allow you to quantify its effect on melanopsin. And following on from that, a couple of years ago now in Manchester, my colleague Tim Brown and Ken Wright from Colorado convened a meeting where we got together a group of experts to give some quantitative guidance for how much light people should be exposing themselves to. And that's been published. And it's a first pass. I imagine that over the years, that advice will be revised. I hope it is, because we should find out more. But at least we can start saying, for example, if you're running a nursing home, this is the type of light that you should be exposing the residents and staff in your nursing home to through the day and at night. And you can buy a light meter and you can say, am I hitting that or am I not hitting that? And I hope things like that are going to have a really quite wide reach, and we'll start having just a more appropriate light exposure for everyone, which will be fantastic.
Yeah, this is really interesting to change light exposure in different settings, offices, hospitals, home as well, right?
Yeah, definitely at home. I mean, most people are fortunate enough to be able to choose their own lighting. So to be able to have a simple meter that you can have in your home and say, I'm having trouble sleeping at the moment. Is that because the light in my house is wrong? Just measure it and say, well, no, it isn't that. Or, yeah, it is. You might want to try that. Try it and see if it helps, those sorts of things.
Yeah, very interesting. And I hope we can see some progress there in the next 10 or 20 years.
Oh, I'm sure we will. I really am.
Great. I think this is a lovely note to end on. So, thank you so much for coming on the podcast. It was a pleasure.
It's my genuine pleasure. Thanks so much, Caro.
Many thanks to Rob for joining us for this episode of Light O'Clock. Today, we've learned about this special cell type in the retina, which contains the pigment melanopsin. These cells are the iPRGCs, and they're responsible for what we call the non-visual effects of light, meaning effects of light on our body which are not related to vision. So, for example, regulating timing of hormone production and effects on circadian system. And while we're talking about hormones, you might have noticed that we have mentioned the word melatonin a couple of times in the podcast. So if you're wondering how light, melatonin, and circadian rhythms all come together, we've got you covered. You just need to tune in into the next episode in 2 weeks, where we'll be chatting to Elise McGlashan. As always, if you're interested to know more about today's topic, you can check out the show notes for relevant links. If you have any questions or feedback, we would be happy to hear from you. You can always reach us at our email, which is listed just at the end of the show notes. Until next time, and in the meantime, don't forget to lighten up your clock.